Hello everyone, my name is Cash, and in this video I'm going over the anatomy of the eye and its functions. So the first thing I want to talk about is the orbital cavity, which is your eye socket. And the orbital cavity is basically everything around your eye that isn't the eye itself. So that's your lacrimal gland, your lacrimal sac over here, and then down further your nasolacrimal duct. Now, the importance of your orbital cavity, outside of the fact that it houses your eye, is that it acts as a sort of defense against the outside environment from getting into your eye. So the components of the orbital cavity help protect against debris getting into your eye. And the first way it does that is through your eyelashes. Now, your eyelashes are more catered toward filtering out heavy debris like hair follicles or large dirt particles and even moisture. Then, for your smaller debris like dust and sand, that's where your lacrimal gland comes in. Now, your lacrimal gland produces tears. So, say you get dust or sand in your eye, your lacrimal gland senses that and sort of like almost like a waterfall will produce tears, and it is like a drainage system almost, because it's the drain is sort of at a angle, so the tears will filter this way. So they will filter down. So the tears will come down, clean all or hopefully all of the particles out of the eye. Then it'll move toward into your lacrimal sac, down into your nasolacrimal duct, into your nasal cavity, where it can be disposed of. The next thing I want to talk about is the tunica fibrosa. And this layer of the eye is made up of three main parts: your conjunctiva, your sclera, and your cornea. Your conjunctiva is a thin layer that surrounds the front of your eye and lines your eyelids. And I'm going to be jumping back and forth between these pictures because I couldn't fit them on the page. But so your conjunctiva is a mucous membrane which is made up of epithelial tissue, and in this epithelial tissue there are goblet cells. And these cells, what they do is they produce mucus. So that mucus is what lubricates your eye, because your eye needs to be hydrated constantly or else you'll dry it out, right? So, and that's why we blink, because your eye is trying to, re-lubricate itself. So, then you have your sclera. Now, your sclera is this outer sort of thick white layer. And when you look at an eye, the white part you see is the sclera. And what that does is it sort of protects against any like puncture wounds, abrasions, anything like that. So it just keeps the inner parts of the eye from physical damage. Oops. Last part, your cornea, is this bumped out region right here. And it's kind of hard to see in an actual depiction of an eye, but what it does is it helps refract light that's coming into the eye, because as we'll get into, your eye needs to refract light in order to process it for the brain to make an image. Now, the next part of the eye which is the tunica vasculosa, is the middle part, and it definitely has the most components of the 3 different layers. So in this layer, you have the iris, the pupil, ciliary body, suspensory ligaments, accommodation, which isn't a part, but we'll get into it, your choroid, your aqueous humor, and your vitreous humor. Now your iris is the little colorful part right in between your sclera. And what it does is it's a smooth muscle, and its job is to control the amount of light that is let into the eye. So when your iris constricts, your pupil, which is the little black entering to the back of your eye, will get smaller. And when the iris dilates, your pupil will get bigger. So when your iris dilates, it's letting in more light. When your iris constricts, it's letting in less light. Now, your ciliary body is a very important structure. Ciliary body contains ciliary muscles, and these muscles directly attach to suspensory ligaments, which you can see are these little white sort of like ropes. And the ligaments are attached to the lens, which isn't listed here, but your lens is like a magnifying glass type structure. So this all leads into accommodation. And what accommodation is your eye accommodating for different distances of light it's focusing on. So when I'm looking at an object that's far away and the light is entering into my eye, it sort of refracts. It is more refracted due to the distance it has to travel to get to my eye. This is important because what your ciliary body does, it senses this. So say I'm looking at something that's far away, ciliary body senses this, it will constrict, which pulls the sensory ligaments, and then as a result, pulling on the lens, flattening it out. And when the lens is flattened, it doesn't have as much refracting power, because you have to refract the light perfectly to the back of the eye or else it'll look blurry. So when the ciliary muscles pull it, ligaments are taut, the lens flattens out, and it doesn't have as much refracting power because it doesn't need it. If it didn't get pulled and it didn't flatten out, the light would refract too early and it would set before it was at the back of the eye. It would reach a focus point before it was at the back of the eye. Now, vice versa, if I'm looking at something very close, your ciliary muscles are going to relax. And when they relax, the ligaments are going to loosen. And if they loosen, the lens isn't being pulled, so it takes its more natural, fatter shape, which has more refracting power because it needs it. Because when you're looking at a close object, the angle of the light is wider, so it needs to refract it more in order to get the perfect focusing point at the back of the eye, or else it would focus it behind, theoretically behind the eye, and it would appear blurry. Okay, next we have your choroid, and what this is, it's directly below the sclera, and what it does is it is very vascular, so it provides blood supply to the different parts of the eye. So you can see it extends all the way around, supplying blood to all these different parts. Also, it contains melanocytes. These melanocytes absorb light, which is very important because you want to minimize the amount of excess light that is entering the eye. Because if you get too much light entering the eye, it can overwhelm the brain and it's not able to process. It's too much information for it to try and process. So you want to absorb any excess light that you can, and that is the function of the melanocytes, is to absorb light. Then you have your aqueous humor, and your aqueous humor fills the cavity, the anterior chamber, which is this right here, between the iris and the cornea. And the aqueous humor is very important because it provides structure to the cornea and keeps it elongated. As you can see, it's sort of bumped out here. This is important because it provides the cornea with the right angle or the right structure to perfectly refract light in the way it needs to. If there were to be no aqueous humor, the cornea would flatten out and it would refract light wrong at a wrong angle, and the eye would not be able to focus it as well. Then you have the vitreous humor, which is much more abundant in the eye than the aqueous humor, and this whole purple area back here is full of vitreous humor. It's kind of like sort of jelly. It's more gelatinous than aqueous humor, which is more watery. So the vitreous humor just sort of provides structure to the eye and a little bit of refracting power, but its main purpose is to just provide structure to the eye. The last layer of the eye is the tunica nervosa. And in this layer is where the beginnings of visual processing occur. So you have the retina, the optic disc, the fovea, and photoreceptors. Now your retina is this yellow band here that extends almost your entire eye. In the retina, there are a series of specialized cells, like photoreceptors, which are responsible for the generation of a signal that is sent to the brain. Now, next you have the optic disc, and the optic disc is responsible for picking up all of the signals sent from the nerve fibers in the retina, and then sending them back to the optic nerve, and then the optic nerve up to the brain. It's important to note, at the optic disc there are no photoreceptors, which means you cannot have a generation of a visual signal here. And that is why we have a blind spot in our vision. It's because at this spot specifically, there are no photoreceptors, so you cannot generate a signal. Then we have the fovea, and that is this little divot here. And the fovea is the center of your retina, and it is where vision is the absolute best. And this is because when light enters your eye, it has to travel when it reaches the retina, it has to travel through all of these structures and then reach the cones and rods. Everywhere else, there's a layer of blood vessels and structural cells, so they absorb some of the light that is sent into the eye, whereas the fovea has the least amount of these filler cells, so light can more directly reach the cones and rods. On that topic, when light is sent down through to your cones and rods, the way a signal is generated is through pigments in the membrane of these photoreceptors. So within the pigments themselves, there's a molecule, and it is called retinol. And retinol, what happens when light reaches it, the energy that it carries, the energy the light carries, changes the retinol. And this change in retinol will cause a membrane potential in the cell, or in the cones and rods, which is then picked up by the bipolar cells, and then the ganglion cells, and then to the nerve fibers, to the optic disc, optic nerve up to the brain. Lastly, you may be wondering, well, why do I need cones and rods? That is because cones are much less sensitive to light itself, meaning you need brighter light in order to generate a membrane potential. Also, cones are sensitive to color. This is because there are three different types of cones due to different pigments present in each of them. So you have red cones due to red pigment in the membrane, blue cones due to blue opsin pigments in the membrane, and green cones due to green pigments in the membrane. And they're all sensitive to these three are sensitive to different wavelengths of light, which we interpret as color. Then your rods, they're all the same. They're all sensitive. There's one type of pigment they have, which is called rod opsin. And this pigment is not sensitive to color. And but it is very, very sensitive to light. So these rods help us specifically in situations where we're in the dark. Because they're much more sensitive to light, they can pick up on much less light. So that is the difference between cones and rods. To summarize this whole presentation, I think we're going to follow a detailed path of light into the eye to understand exactly what happens in the eye itself when light is picked up. So, in this situation, we'll say we're looking at a faraway object. So, the light, already somewhat refracted, enters the pupil here. The pupil refracts it even more into the lens, which in this situation is flat, as depicted here. Refracts the light, the lens refracts the light. It's focused at the site of the retina. And in this situation, it's pretty bright, so we'll say that mostly cones are activated. The cones generate a membrane potential due to the change in the retinol molecule in the pigment of the membrane. This generates a membrane potential picked up by bipolar cells, generates another potential into ganglion cells, travels the nerve fibers, sending to the optic disc, back the optic nerve, and then up to the brain. So I hope this maybe helped a little bit in allowing you to understand the process that goes on in the eye. It's a very complicated thing, and it's kind of hard to grasp, but I hope I maybe sort of made it a little bit more understandable. And yeah, so thanks for watching.